So what we'll do right now is look at what happens when there are two different characteristics being looked at the same time and being crossbred. So the type of experiment that Mendel did was then choose two different true breeding characteristics. So in this case, let's say tall plants versus short plants. So you can have tall plants breeded with tall plants and get tall plant offspring. And then those would be bred with one another, and their offspring would all be tall 100%, so they were true breeding. And then the same thing with short. You can get some short plants, breed them, short offspring, and so forth. Take the true breedings that were tall and short and breed them together. All the offspring are tall. So tall becomes the dominant. Right? So that's what we have here. So tall is going to be the dominant. And short will be the recessive. In our previous example with a monohybrid cross, I was looking at or talking about flower color, and we looked at red versus white flowers, with red being dominant. Okay, in this case, so red was the dominant, and white is the recessive. So the idea here is, if you were to look at then two traits simultaneously, or more, but right now we're just going to look at two. Is there a particular pattern? Like do dominants always go together, or recessives always go together, or is there something like that could be explained, or is it more of a random distribution? And so the idea would be to create plants that would be tall and red, and tall and short, and tall and white, and short and white, and so forth. And then try to breed them and look at all the different patterns. And it's a whole variety of different complex experiments that would be done to look at all the offspring and count them out. But what we're going to look at here is then what would happen if we just took the parents who were tall, red, okay, so dominant, okay, and then short, white, the recessive. So for the tall ones, we're going to have T capital, because they're both dominant, and this is going to be for true breeding parents, okay? And then the red, obviously also going to be the true breeding, so they're the same. Short, same thing we have here. Short, sorry, that's the T's. So the short is the lowercase t, and then the white is the lowercase r. Now the thing is, if we mate them with one another, these individuals, when it comes to this particular gene, have two choices, the tall or the tall, which really means only one choice, so they can only give to the offspring the tall gene. Since tall is dominant, the other mate can give the short gene, and that's it, there's no other option. It doesn't really matter, they're all going to be tall in this first generation. So this is the parent generation, and here's an F1 generation. Here, all tall. And then we look at the red, the same thing would happen. This individual can give the gene for red flowers. This one can only give the gene for for white flowers, and so they're all going to be red. Okay, so they're all tall, all red. All right, and so they would be this heterozygous genotype and the tall red phenotype. Now what we would do is then take these and cross them to get an F2 generation, and that's where it starts to get much more interesting, all right, to see what happens. In the F2 generation, now there are some more options. So you have two of these being crossed with one another, right? And so this particular individual can give the dominant for tall and potentially give the dominant for red. It could give the dominant for tall, or it could give the recessive for white. So either way. And then if we flip it the other side, also 50-50. So tall, and then also 50-50, it can give the short gene. then when it comes to the color of the flowers, it could also still give the dominant red, or it could give the recessive white. And then the other individual that it's mated with has all the same options. Okay, so it can be homozygous, where both are going to be dominant. It can give both recessive, or it could have combinations of the dominant with the recessive for the two different genes. And that's speculation, but then we would have to breed them and actually look at the offspring and see, based on their numbers, if that's really true or not. And what Darwin then found out is it was true, because, well, look at this table and how it fills out, and then look at the numbers and how they would fill out. So bear with me for a second as we fill out, the table here. Going to have the tall, red, tall, and then this one will still be red. Tall, red, and still tall, red. So these are all tall red, which isn't that surprising because the dominant genes are going to be more prevalent in terms of their phenotype and being observed. Tall, again, red, tall, and now we have a tall white. So this one is going to be, we'll note here, something different. Here we're going to have tall again, back to tall red. Sorry, there we go, tall, and this one will be tall white. So now we see in this particular generation, or not generation, but this particular column here, sorry, tall red and tall white, 1-to-1, or 50-50. Keep moving along. So again, tall, red again, tall, red again, and now see the first short one here, short and red, and I think I have another color here. And then we have, again, short and red. So another one here. Drop that down. So, still tall reds in this group. But now we're going to also see some short red. So in this particular case, the recessive for the color appears in this column here, and the recessive for the height appears in this column. And it's the same thing, it's 50-50, or it's a 1-to-1 ratio. Yikes, okay. Keep moving along. So we got the tall again and red. Okay, tall again and white. Same as we've seen over here. We're going to have now short and red. I lost my, there we go. Like we saw. And then we're going to have here short and white. So now in this final column over here, we actually see 4 different phenotypes, right? So here we have tall red, here we have tall white, we're going to have short red, and then short white. And they would be equal, 1 to 1 to 1 to 1. If you were then tallying up the whole thing, so there's 16 boxes here, tally up how many tall reds there are, and we go through and count them all, we find out of the 16, there are 9 that are tall red. There are 3 that are tall white. There are 3 that are short red. And then there's 1 that is short white. Now, you also note that I did something a little bit different here. When I told you the phenotype, the appearance, okay, so again, the tall red, tall white, I was writing genotypes below them. Short red, short white. And you'd notice that for some of them I completed it and others I didn't, and I just left dashes, right? And that's because that's where we will start to get into when you work out some genetics problems and try to solve for, say, what was the genotype of the parent if these were the offspring, and then try to figure that out. What you would know is you'd be given a particular phenotype of offspring, and you could then know certain things. You could know that, oh, well, the parent must have at least one of these alleles because we see the dominant trait. We don't know then necessarily if they are homozygous dominant or heterozygous dominant because we're just looking at their appearance, their phenotype. We do know we have one of those genes. We don't know if we have the others. So what we'd have to do is cross them with other individuals and then find out what are the proportions or ratios of their offspring. If they're all tall, we would know that it was homozygous for tall. If some of them are short, then we would know it was heterozygous because it had the short allele, so some of the offspring would be short. And those are separate things we could go over when I go over some actual problems. Now for the short white, there's really no option because they are recessive. They have to be homozygous, so those individuals must have this particular genotype, and you would know that for sure. So whenever you see the recessive, it has to be homozygous because otherwise you wouldn't see in phenotype, in appearance, it being expressed. Whereas with the dominant trait, you don't know when you see it in the phenotype if the organism who displays that phenotype is homozygous or heterozygous for the trait, unless you look at the offspring, and a lot of offspring, because then you could collect some data on it. Now, what does this tell us about our broader discussion? Okay, because the broader topic really is not, for our purposes, doing Punnett squares. Our purpose is looking at, again, genetic diversity in the population, and something that then Gregor Mendel referred to as law of independent assortment. So what is this, and how is this related to the law of independent assortment? What's being said here is that, see, when you get here to this heterozygous individual, that for these two separate characteristics, height and flower color, that they assort independently, meaning that when gametes are formed, this parent here can form gametes that contain both the dominant alleles. They can form gametes that contain both the recessive alleles, or they could form gametes that form combinations of the dominant and recessive alleles. So they're independent of one another, right? It's independent assortment, equally possible that you could have any combination of them. What we're going to do next is then look at the chromosomes, and we're going to go back to meiosis. And in meiosis, show how that independent assortment actually works from the chromosomal aspect of it. And we'll go through it in two ways. One is just straight chromosome, and then look at other ways that we increase genetic diversity through a process called crossing over. So for this, you should be able to work out a basic Punnett square, look at the ratios, and kind of explain that, and also you should be able to see how that relates to this independent assortment, or how independent assortment is demonstrated through something like this, with the idea that two different traits, where we look at segregation. So keep in mind, segregation, we're talking about a single, I'm blocking it from me, but not from you, a single characteristic, so that the dominant or the recessive is equally likely, all right, and that that's the segregation we're talking about. With independent assortment, we're talking about two different characteristics, and that they are also independent from one another. So next we'll go to the meiosis view of this, and how it's all explained in the chromosomal patterns.